Dear students, Assalamu alaikum. Today we are going to talk about our course special topics in biochemistry, course code CHEM 6462. This is lecture number 1 of our online classes. The topic of today's lecture is contemporary biochemistry and molecular biology techniques. The outline of today's lecture consists of polymerase chain reaction, or it is abbreviated as PCR. Then we will very briefly talk about gel electrophoresis, because in subsequent lecture I want to talk separately about this, because this is a very important topic. So we will talk about gel electrophoresis briefly, and then we will talk about briefly about quantitative real-time PCR or QRT PCR. Then in the subsequent lectures we will talk about immunohistochemistry and immunocytochemistry. I have selected these topics in a sequence because these techniques, they corroborate each other's results, and they are very important for understanding the results of each other. So now going to our lecture, PCR, a polymerase chain reaction. It provides an extremely sensitive mean of amplifying a relatively large quantity of DNA. It was first described in 1985 by a scientist named Kerimoulos, and he got Nobel Prize for it in 1993. Since polymerase chain reaction was introduced into the fields of biochemistry, molecular biology, it has brought numerous advances in understanding various aspects of these subjects, and it has gained enormous importance, and it can be termed as a momentous event in the history of molecular biology, because after that many techniques or many things that were very difficult became easy and solvable through using PCR. So this is why it is very important. Now first we talk about the materials that are needed for PCR. Among the material that are required, we require template DNA. What do we mean by template DNA? It means the DNA that is to be amplified is required. We require DNA nucleotides. These nucleotides obviously work as building blocks for new DNA, and they work like, for instance, bricks would play their role in the construction of a building or a room. So similarly, when the DNA is amplified, we require DNA nucleotides, because these nucleotides will function as the building blocks for the newly formed DNA. Then very important players are primers. Primers are single-stranded DNA between 20 to 50 nucleotides long, and what they do is that they anneal with the parent strand, and then DNA polymerase can add free nucleotides complementary to the parent strand in the daughter strand. But before DNA polymerase can function, they need to add they need to have these primers, because it functions as a starting point for the polymerase. Then we require DNA polymerase enzyme, which function in the synthesis of new DNA. Alongside these, we require a buffer. Provide environment for the for carrying out these reactions. And then we require a magnesium ion. It functions as a cofactor for the functioning of the DNA polymerase. So overall, the slides consist of all the materials that are required for conducting a successful PCR reaction. So before doing a PCR reaction, we need to have these material in hand. Then there are three major steps in the process of PCR. One is denaturation. It means first the DNA that is to be amplified is denatured. Then is the process of annealing. Annealing is the situation in which the primers anneal with the parent strand of the DNA. And then is the phase of extension. Extension is the phase where when after the primer attachment, the DNA polymerase is provided with the environment to be able to add complementary bases to the parent strand. So these three are the major steps of conducting a PCR reaction, and we are going to talk about this in greater detail in the coming slides. Now, this is a cartoonic representation of how the polymerase chain reaction functions. Now, this is a template DNA, and this template DNA is to be amplified. Now, through when the reaction mixture is put in the thermocycler. Thermocycler is the machine which varies the temperature according to a programmed way. In the first step, in step 1, that is called denaturation, for 1 minute it will take the temperature to 94 degrees C, and what will happen as a result of this process, it will denature the template DNA. What is meant by this denaturation is that the two strands of the DNA, they will be separated from each other like this. You can see they are separated from each other in the first step. This is called denaturation. This is important because for the annealing of the primers, we need to have this denaturation, because primer can only anneal with a single stranded DNA. Now, the point is, you know that there are two types of bonds between the DNA. One is the bonding that is between the nucleotides from one nucleotide to another, and another type of bonding is the hydrogen bonding between the different bases. So as you know that hydrogen bonding is relatively much more weaker than the bonding between the nucleotides themselves. So the weaker bonds, that is the hydrogen bonding, is broken, and the two strands are separated from each other. Now, in the step number 2, that is annealing, the thermocycler will bring down the temperature from 94 degrees C to 54 degrees C for a period of 45 seconds, and as a result of that, what will happen is that the primers which are in the reaction environment, they will attach with their relative complementary sequences. Like you can see here the primers that is 20 to 50 nucleotides long, they attach with their complementary sequence, and here also they attach with their complementary sequence. And once these primers are attached, you have to keep in mind that forward primer and reverse primers, they attach in such a way that, for example, if this is the 3' to 5', and the primer here will attach from 5' to 3', and as you know that DNA is anti-parallel. So, and here also 3' to 5', the primer attached in the 5' to 3' fashion, in anti-parallel fashion. Now, once the primer are attached, the thermocycler will raise the temperature to 72 degrees C for about 2 minutes. Now is the situation when the DNA polymerase will act. It will start to recruit these freely roaming nucleotides. But on what basis will these nucleotides be recruited? It depends on the parent strand. The nucleotides that will be recruited will depend on the sequence of the nucleotide in the parent strand. For example, if here it is A, it will recruit T in the daughter strand. If here it is G, it will recruit C in the parent strand. If it is, say, T, it will recruit A. If here it is say C, it will recruit G in the parent strand. So complementary to the parent strand, it will start to incorporate the nucleotides. And here is an animation of how this process takes place. One can see that the three processes, they distinctly happen. The denaturation denatures the DNA, then primers attaches the RNA, the DNA polymerase start to lengthen the sequence, and then after again the cycle is repeated, 94 degrees C, the strands are separated, the DNA polymerase comes in after annealing, and it adds nucleotides to the sequence. And once the cycle is completed, again the cycle repeats itself for 94 degrees C, and then 54 degrees C is the annealing temperature, and then it moves to 72 degrees C to raise the temperature and start the process of DNA polymerization. So here, how is the it shows how in real time and the process of denaturation, the process of extension, and the process of annealing, these three processes, they take place, and how the temperature varies for different processes, and how the DNA polymerase functions. So hopefully this little animation is very helpful for you to understand how this process occurs in real time. Now what happens is this, that for example, this is the template DNA. In the first cycle we see that these two strands separate, and for instance, if this is the parent strand, this would be the daughter strand, this is the parent strand, this would be the daughter strand. So we see that exponential increase in the number of copies. In the second cycle, you see these two will be converted to another two strands. So one is this, one is this. This will be converted to another two, and this will be converted to another two, and this will be converted. So now you can see that as a result of these different cycles, we have 4 copies, 8 copies, 16 copies, 32 copies, and overall the process of PCR, it happens for about 36 cycles. So as a result of 36 cycle, this exponential amplification of the PCR will result in 2 raised to the power 36 is equal to 68 billion copies of the DNA. So this is how this amplification of the DNA will take place in such a exponential manner. So this is how so many copies of DNA are produced. Now once the process of PCR is completed, the size of the DNA or the conformation of the reaction can be done through the help of gel electrophoresis. In gel electrophoresis we have a ladder that is a known sequence of a DNA that we know, and this ladder, this is a 3 kb ladder with its one of its band is around like 300, another 500, 1 kb until 3 kb. So it shows the by comparing with the known, we know the different sizes now. For instance, these are the unknown samples that we have done. We see that, for example, this one band and this is another band. We can compare with the ladder, and then we can confirm the size, and we can also see that the reaction has been done. In this situation, we can't see any bands, so showing that either the reaction didn't take place or it was not successful. So in this case we can see also a very clear and sharp band. In this case we are seeing 3 bands. So this is how the verification of PCR product can be done on agarose or saccharide gel. So these are different PCR fragments. So this is how, but this is how this gel is prepared, how the reaction is done, the gel is run. I will talk about this in a separate lecture, but hopefully you get idea that once the process of the PCR is done, so many copies are produced, the product of the PCR is visualized by the help of gel electrophoresis. Now, one important aspect of conducting a successful PCR is designing of primer. So there are some important points that needs to be kept in mind while designing primers. What they say is perhaps the most critical parameter for successful PCR is the design of primers. This is very true, because primers design, if it is not according to the protocol, it can lead to the failure of the PCR reaction. So there are critical variables that has to be kept in mind. The primer needs to be specific to their the sequences they would need to. Primer length is very important. Primers should not be very short and they should not be very long, because if primers are short, they can unspecifically bind to a sequence that they are not aimed for to be bind with, or if the primer length is very big, it will be difficult for the primers to anneal with the sequence they intend to anneal with. So this is why primer length is very important. Complementary primer sequences, they are important. GC content is very important. As you know that between 80 there are 2 hydrogen bonds, and between G and C there are 3 hydrogen bonds. So GC hydrogen bonding is much more stronger as compared to AT or TA. So this is why primers should have a significant amount of GC content, because this will help for better annealing of the primer to their complementary sequence. And then 3' sequence is important. The melting temperature of the primer is very important. So these are the different parameters that I have put it for you to look at in greater detail, and I hope that these different points, they are self-explanatory, and once you read it you will be able to understand it. However, if you have any question, I will be very happy to answer this. Because of the time shortage, I will not be able to discuss all these together, but you can read it and then we can talk about it if you have any doubt about this. So there are different types of PCR machines that we have. One is the real-time PCR, or the quantitative real-time PCR. Then we have touchdown PCR, we have gradient PCR. So there are different variations of these PCR machines. Now, what we do messenger RNA levels tells us is, from, as you know that from DNA we get messenger RNA, and from messenger RNA we get protein. So messenger RNA level tells us, it reflects level of gene expression. It gives us information about cell response. It gives us information about protein production, which is not always true, because sometimes this messenger RNA, before translating into any kind of protein, is degraded or is utilized. So this is not always true, but it can, in most of the cases, it can give us this these information. Now this is a quantitative real-time PCR, which this is from one of our published paper, which shows that there is a one transcription factor. It is a result of removing a certain stimulus. It increases the its expression several folds. These stars represent the statistically significant expression as compared to control in day 1 and day 7. So this is it is showing that real-time PCR we have measured a certain transcription factor whose expression increases by several folds as a result of removing a certain stimulus, and then it comes down to the normal level, showing that this is a transitory expressed transcription factor. But this is how through the help of PCR we can measure the expression of different proteins or different genes. Sorry about protein. So different measurements can be done for different situations. Now there are numerous applications of PCR. One of those important applications is DNA sequence technology. It means that with the help of this technique we can sequence DNA. Isolation of a particular gene from genomic DNA can be done through this PCR, and then, as you saw in today's lecture, amplification of DNA can be done with the help of PCR, and then as you know, the diagnosis of different diseases can be done with the help of PCR. Mutational analysis can be done by comparing the sequences, and quantitation of messenger RNA in cells or tissues can be done, and it can be used in forensic medicine, that is in crime scene investigations. In quantitative real-time PCR is also used for quantitation of gene expression. It can be used for drug therapy, efficacy, or drug monitoring. It can be used for viral quantitation. It can be used for pathogen detection. So these are the different vast range of applications of the different PCR techniques. Here we at the end of the lecture we take a case study of a particular situation where how crime scene the PCR technology can be used. So we try to understand how do crime scene investigators create a DNA profile. So now these are the possible evidence that can be collected from a crime scene. That can possibly be blood, it can be teeth, it can be semen, it can be hair, it can be urine, it can be bone, it can be tissue, so it can be saliva. So these are the different crime scene evidence that can be collected from the crime scene. And sorry about that. So these are the different samples that can be collected from crime scene. And now for crime scene cadets, here's your case. Use DNA profiling to determine which suspect cannot be excluded from suspicion. Now, as you know that once these different samples can be collected from the crime scene, as you know that every crime has a possible suspect. So what we do is that we isolate the genome of these from these samples, and then we take the and we run it on the we do the PCR and we run it on the gel. So the one is the first one. This is the allele ladder. This is the ladder. This is the sample which was collected from the crime scene, CS. From the crime scene, we are getting these two bands, and these are suspect A, suspect B, suspect C, and suspect D. If we run the genome of the DNA of the suspect A, give us these two bands, suspect B, give us these bands, suspect C, suspect D. Now, once we have these two bands on the suspect from the crime scene, and then you compare it with that of suspect B, we see that these bands are matching, while A, C, and D, they are not matching with the one from the crime scene. So in this situation, suspect B cannot be excluded, and it has the sample that we collected from the crime scene and that of the suspect B, they are matching with each other. So suspect B cannot be excluded while the others can be. So it shows that the culprit for this particular type of crime is suspect B. This is how the crime scene investigators, with the help of DNA profile, can reach to the culprits of different crimes. This is one example. There are many others. I hope you enjoyed the lectures, and if you have any question, I would be very happy to answer you. Please let me know about your feedback. Thank you very much for your attention. See you in the next lecture.